Hello dear learners, in our today's class we are going to learn about a technique which is referred to as ELISA, or in full form it is also referred to as enzyme linked immunosorbent assay. So first of all we should know that what is the meaning of assay. Assay is actually a kind of test which we are doing. Either it can be related to the confirmatory test, or it can be the concentration based test, anything like quantitative or qualitative. So that is assay, and here we are doing the immunosorbent assay. So it is basically immunity related, we know this, and it is antigen antibody interaction. Enzyme linked. Now what is the meaning of this? See here, the term enzyme linked is the most crucial thing. Let us understand that what do we mean by enzyme linked. In the whole of the ELISA, we generally do one thing, that is, we take one antibody, one on the other form of antibody, or even we can take the form of antigen which is linked to an enzyme. And the whole ELISA is based on the color. The whole test is based upon the observance of color. So when we add the substrate to this enzyme, the enzyme and substrate will combine and will react to form a product. Let us assume the blue colored substrate I am adding to this enzyme. So this enzyme will convert this substrate into product, and the product will be a colored product. So in my particular well, I will observe the color. If I have firstly the enzyme linked antibody or enzyme linked antigen, and secondly if I have substrate. So that is much important to be understood. Now this particular interaction will produce the color, and on this color production is our whole ELISA based. So the main principle of ELISA lies behind the antigen and antibody interaction, and either of them will be having an enzyme, and when we add the substrate to this enzyme, so we can say that the color will be produced, and it would be produced only if this antigen is attached to this antibody. Otherwise this would have been washed off. So washing is also very important in ELISA. So now on basis of this, let us try to understand the types of ELISA. We have four types of ELISA on the performance basis. The most important and fundamental being the sandwich ELISA. So we are first of all we will try to understand the sandwich ELISA and its working. Now suppose there is a patient, and the patient is suffering from a disease. There is a patient and he is suffering from a disease or an infection, and doctors say that there might be a pathogen X, which we can say that there is an antigen X present in the patient. Now to confirm the presence of that antigen, we perform ELISA. So it is a kind of assay. So in the ELISA assay, what do we do is that we have to confirm whether this antigen X is present inside the patient or not. So try to understand the procedure. First of all, because right now we are doubtful that whether the antigen X is present or not. So what we are going to take, we are going to artificially take the anti-X antibodies, because we have to just check whether the X antigen is present or not. So lab created or from any other animal, if we are obtaining this anti-X antibodies, we will take them, and then we will take the serum of the patient. Now serum of the patient will definitely contain the antigen X if it is present inside the patient. So how do we start with the sandwich ELISA? The process is very simple and I have to explain you. Let us zoom into this. What do we do is, as I told you, the requirements were very clear to you now. In this, first of all, I have to take the anti-X antibody, which is normal anti-X antibody, and I will coat that here in the well. Now this particular is a well. A well is a type of test tube, but in a very micro form, where we are going to perform the experiment. Now after this anti-X antibody is coated means fixed inside the well. Now, it means even after washing this should not get removed. The second important thing will be that now we have to add this particular serum which may have this antigen X. So suppose let us assume that this is the serum of patient and this is the antigen X. Let us assume that antigen X is present. Now what should be done? So this serum should be taken out and there should be emptied inside it such that these antigens, I am not drawing this liquid here, I am just adding these antigens such that these antigens if present are added. Now if the antigens would have been present, they would definitely bind to their specific antibodies which we already have coated inside the well. So in almost all the antibodies what happens now is that the antigen from the serum gets coated. So here we have the serum and the antigen. For your satisfaction I should also draw this, that this is my serum here. Wait, okay, so here is my serum and in that the antigen was present, so I can mark this is the antigen. Sorry, I should again zoom it more. So this is the antigen which came out from the patient. Now what I will do now is the most important step. I will add another anti-X antibody only again. This is also the artificial one or it is obtained from any other uh, animal which is compatible or whose serum and whose antibody is compatible to the antigen X. So this anti-X antibody, but this is linked with an enzyme. Linked with an enzyme. So as you can see, this is our enzyme. Now what we are going to do, this particular antibody we are going to add inside this particular thing. As soon as we are adding this whole sample, what happens is that this antibody, because it is specific to antigen X, it will bind to antigen X. So similarly all the antibodies, means the secondary antibodies, will be binding to the antigen X. So we will also label them as primary antibodies and secondary antibodies. Primary antibodies are those which were firstly coated inside the well. So I can say that these are my primary antibodies, these are my antigens, and these are my secondary antibodies which are linked with the enzyme. The next step would be the addition of a substrate. Suppose we are adding a substrate which is very specific to that enzyme, because we have created that secondary antibody and we know that what enzyme is there. So we also should know that what substrate should we add so that the color can be produced. Okay, in between that we also do the washing so that all the leftover debris should be removed, and if the antigen was not present then the secondary antibody should also have been washed off. If the washing off is done, now if I am adding the substrate, so let us try to add this substrate here. I am taking the substrate adding here, then this should happen ideally if the antigen was present. Okay, so here the substrate is binding with the enzyme. This way the substrate has bound to enzyme. I am not drawing on the last because the space is not available. As soon as I have bound this substrate particularly, now what happens, because now the product has to be formed, the product forms and the product formation is colored. So I will see a colored product being formed. So let me write it like this. Okay, so this is my colored thing which is formed. Now I'll again wash it. After washing also, if the color is fixed, it means the antigen was present onto which the primary and secondary antibodies got fixed, and because the primary and secondary antibodies got fixed, the color was produced. As simple as that. Now if I need to plot the graph for this, what kind of graph should I obtain? The graph should be with respect to what? The graph should be with respect to the absorbance. So wait, this is very bold. If I'm drawing this as x and y axis, and here if I'm plotting the graph should ideally be like this. This should be my graph. Why like this way? Because, and also there should be a saturation point, because no more further antigens are present, so no more further antibodies can be added. So this is what my saturation point will be there, because, and this saturation point will definitely tell me the concentration of antigen. Here I can have the concentration of antigen also, but no, because this is not the optical density graph, I cannot say. This is just the time being with time, and this is the, let's say, absorbance, or let's say the color produced. So color is produced, and with the time the color is increasing and then reaching a saturation point, it means that I definitely have here the antigen present. So this is one of the very important techniques through which we can determine whether the patient has that antigen or not. Moving on to the next one. So the next is next is direct ELISA. Direct ELISA is quite simple. What you need to do is that you have to take first of all the patient, and you have to take its serum. If the serum has the antigen X, what do we do is we take that serum and we try to fix it in the well. Fixing is required because so that the antigens could be coated in the well. Now as soon as the antigens are coated in the well, and if we are if we are suspecting whether antigen X is present or not, so we will add the particular anti-X antibody, which is enzyme linked. So here no primary and no secondary antibody, directly the enzyme linked antibody is used here. So these are my enzyme linked antibodies. See, after each step washing is required, so that to remove the debris and to remove those antibodies which have not bound. So in this way, now for the sake of simplicity, I can, okay, let us leave it like this. Now I'll add the substrate. So here I have added the substrate, here I have added the substrate, here I have added the substrate. As I have added the substrate, it has bound to its own particular enzyme. And what do I see? I see after a certain period of time that there has been a color production here which has been observed like this. So this is the case where I can clearly say that the antigen was present, and because the antigen was present, the antibody has bound to that antigen, and because it has bound, the substrate bound to the enzyme, and even after washing, because the color did not go off, that definitely means that there was antigen present. So this is a direct form of ELISA. See, direct form of ELISA and indirect form of ELISA, if I compare specificity, so specificity is the sandwich ELISA, because in sandwich ELISA we have used two antibodies, primary antibodies and secondary antibodies, but in direct ELISA we only use the enzyme linked antibody. The specificity is low, might be possible that due to other reasons also the antibody got coated itself into the well, and even if there was no antigen present, but still we are finding the traces of color. So the specificity is low, so the results are obviously poor as compared to what the sandwich ELISA is producing. Now let us move to the next form of ELISA, that is the indirect ELISA. So indirect ELISA is again very important. Here what we are doing is, here we are trying to confirm the, see this is different, because here we are now confirming what? We are not confirming the antigen X, we are confirming anti-X AB in the patient. This time it is different. It is just reverse. We have to check whether an antibody particular to that antigen has been produced in the patient or not. If the antibody is there in the patient, that means particularly the antigen should also be there. So this time we will use the patient's antibodies, and then we are going to add our antigen, that is we have taken from outside, not from the patient. So suppose I have added this particular antigen here, and then I have added this particular antigen here, and then this particular antigen here. Okay. After this, because now they will be fixed, we will add our secondary antibody, and this time the secondary antibody, what does it do is, now you have to focus it carefully. The secondary antibody will attach itself like this way, like this. Now what happens here, we have to look into the details. See, from the basics of immunology we know this, that this part is the FAB of the primary antibody, and means from the patient only, and this is the FAB of this antibody, right? Now what is happening is that FAB of secondary antibody is binding to this part, and this is the FC, that is the constant region, that part of the primary antibody. So here the FAB of secondary antibody is binding to the FC region of the primary antibody. And now because it is enzyme linked, so in case if we are adding the enzyme, so here we will add the enzyme, here we will add the enzyme, and here we will add the enzyme. As soon as we are adding, sorry, substrate we are adding, so as soon as we have added the substrate to the enzyme, the color will be formed, and then we can get the presence of the particular antibody. So here we are directly detecting the presence of antibodies in the patient's serum. Last but not the least is the competitive ELISA, and this is very very important. It is totally based upon radio labeling. So let's see what do we do here. See, again here we have to detect one thing, that is we have to detect the antigen X in the serum. Again the normal kind of ELISA only. So basically you look at the two antigens. The two antigens are very similar. Even I should, for the sake, I should try to wait. I will duplicate this only. This is the normal antigen and this one is the radio labeled antigen. See, I am writing here. This is your radio labeled antigen X, and this antigen X we have made or created, or we have taken from outside. Okay, we have taken it from outside and then we have artificially radio labeled it, and this is the antigen X, and this antigen X is from the serum of patient. Now what do we do? First of all, we add the radio labeled antigen to our both control as well as test wells. So I am adding this whole here. Okay, I am duplicating it because now it is added everywhere. Okay, and then same goes for here also. I take it here, 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 and here. So now what I am seeing is that my whole of right now in both of the cases, here also I am getting 100% fluorescence, and here also I am getting 100% fluorescence from these radio labeled antigens. Now what I do is in the test one, now I will add the serum. Okay, here I am adding the serum. Now there is very important thing. What will happen is that as soon as I am adding the serum, so this is my serum. Now let me zoom it out. So this serum now I'm adding here, which is containing the particular antigen. Now the antigens must be in excess. Let's say there are 3 of them here. Now what will happen is there would be a competitive binding or a competition for the binding between the radio labeled antigen and between the normal antigen. And what would happen is this comparatively being weaker will be eliminated and this would be added here. So that is the whole principle behind it. So this particular antigen will be removed. Even after washing, it will definitely be removed. Similarly, this would be removed and this would also be removed. Let's assume that these 3 are being removed and these antigens from the serum would bind here. Now we will do washing. In washing, all the removed and detached antigens and all the debris will be removed. Now here you observe that there is 100% fluorescence, but in comparison here now there is no more 100% fluorescence. Here we have only, let's say, 30% fluorescence. So on the comparative basis you can say that here the actual antigen X was present, and because it bound here, so the fluorescent antigen had to be removed, and because the fluorescent antigen was removed, the fluorescence overall had decreased. So this is what we call as the competitive ELISA. So there we have to compare it, and the competition is actually between the normal antigen X from the serum, and then there is this fluorescent or this radio labeled antigen which we have artificially created. Now if I try to draw the graphs, so all of them follow a similar kind of graph. Here also this can be duplicated for this one, the direct ELISA, because in direct ELISA also we have to mostly see the color, the production of color, right? Now let me take this for here also. In this case also I can say that the same graph is applicable, but in the competitive ELISA the graph will change for sure. So even if I'm doing this, wait, if I try to draw this here, so now my graph for competitive ELISA will change, because here what is happening is with the time the fluorescence has to decrease. And why it has to decrease? It has to decrease only because the normal antigen is bound and then radio labeled antigen is removed. So with the time my fluorescence will decrease. So here I can say because the normal antigen has bound, so after washing, see, washing is very important, otherwise detection cannot be possible. After washing, the radio labeled antigen X removed, and because it is removed, the overall fluorescence decreases with time. So these are the four types of ELISA and whole of the concept and principle of ELISA.